Welcome to Biology, the scientific study of life. The critical reaction, the membrane depolarization, lasts for only a minute or so, thus functioning only as a short-term block to polyspermy. However, fusion of egg and sperm plasma membranes also triggers a series of changes in the egg that cause a longer-lasting block. Sperm binding appears to activate a signal transduction pathways involving two second messengers IB3 and DAG that cause calcium Ca to the power 2 positive to be released from the egg's endoplasmic reticulum ER into the cytosol. The Ca to the power 2 positive release from the ER begins at the site of a sperm injury and then propagates in a wave across the fertilized egg. Within seconds and high concentration of AC or Ca to the power 2 positive brings about the cortical reaction fusion and the exoplasma membrane of numerous vesicles lying in the egg's cortex, the area just beneath the membrane. These cortical granules formed during oogenesis now release their contents into the brevity line space, the space between the plasma membrane and the vitelline layer. Enzymes released from the cortical granules degrade proteins holding the vitelline layer to the plasma membrane, while mucopolysaccharides produce an osmotic gradients, drawing water into the brevity space and swelling it. The swelling pushes the vitelline layer away from the plasma membrane, and other enzymes harden the vitelline layer. Different enzymes clip off and release the external portions of the remaining receptor proteins from the vitelline layers. These changes transform the vitelline layer into the fertilization envelope, which resists the entry of additional sperm. The fertilization envelope and other changes in egg surface function together as longer-term slow block to polyspermy, although studied in greatest detail in sea urchins. The cortical reaction also occurs in vertebrates such as fishes and mammals. Activation of the egg. Another outcome of the sharp rise in Ca to the power 2 positive concentration in the egg's cytosol, in a substantial increase in the rates of cellular respiration and protein synthesis by the egg cell. With these rapid changes, the metabolism the egg is said to be activated, although the binding and fusion of the sperm and triggers for egg activation, sperm cells do not contribute any material required for activation. Indeed, the unfertilized eggs for many species can be artificially activated by the injection of Ca to the power 2 positive or by a variety of mildly injurious treatments such as temperature shock. This artificial activation switches on the metabolic responses of the egg and causes it to begin developing by parthenogenesis without fertilization by sperm. It is even possible to artificially activate an egg that has had its own nucleus removed. This finding shows that proteins and mRNA as present in the cytoplasm in the fertilized egg are sufficient for egg activation, while the metabolism of the activated sea urchin egg increases the sperm nucleus within the egg starts to swell. After about 20 minutes, the sperm nucleus merges with the egg nucleus, recreating the diploid nucleus of the zygote. DNA synthesis begins and the first cell division occurs after about 90 minutes. In the case of sea urchins and some frogs, the events and timeline of fertilization sea urchins are summarized in this table. Fertilization in other species shares many features with the process in sea urchins. However, the timing differs with species and the stage of meiosis the egg has reached at fertilization also varies among species. At the time of release from the female sea urchin, eggs have completed meiosis. In other species, the egg are arrested at a specific stage of meiosis. Upon fertilization, meiosis is quickly completed along with many of the events already described. Human eggs, for example, are arrested at metaphase of meiosis second until they are fertilized in the female reproductive tract. Fertilization in mammals. In contrast to the external fertilization of sea urchins and most other marine invertebrates, fertilization in terrestrial animals, including mammals, is generally internal secretion. In the mammalian female reproductive tract, alter certain molecules on the surface of sperm cells and also increase sperm motility. In humans, this enhancement of sperm function requires about 6 hours of exposure to the female reproductive tract. The mammalian egg is cloaked by follicle cells released along with the egg during ovulation. A sperm cell must migrate through this layer of follicle cells before it reaches the zona pellucida, the extracellular matrix of the egg. One component of the zona pellucida functions as a sperm receptor binding to the complementary molecule on the surface of the sperm head. Binding of the sperm head to receptor molecules induced an acrosomal reaction similar to that of sea urchin sperm. Hydrolytic enzymes expelled from the acrosoma enable the sperm cell to penetrate the zona pellucida and reach the plasma membrane of the egg. The acrosomal reaction also exposes a protein, the sperm membrane, that binds with the egg's plasma membrane. As in sea urchin fertilization, the binding of a sperm cell to the egg triggers changes within the egg that lead to a cortical reaction, the release of enzymes from cortical granules to the outside of the cell via exocytosis. The released enzymes catalyze alternation of the zona pellucida, which then functions as the slow block of polyspermy. There is no known fast block to polyspermy in mammals. After the egg and sperm membrane fuse, the whole sperm, tail and all, is taken into the egg, which lacks a centrosome. A centrosome forms around the centriole that acted as the basal body of the sperm's flagellum. This centrosome, which now includes a second centriole, duplicates to form two centrosomes in the zygote. This will generate the mitotic spindle for the first cell division. The haploid nuclei of mammalian sperm and egg do not fuse immediately as in sea urchin fertilization. Instead, the envelopes of both nuclei disperse, and the two sets of chromosomes, one set from each gamete, share a common spindle apparatus during the first mitotic division of the zygote. Thus, only after this first division, as diploid nuclei from in the two daughter cells, do the chromosome from the two parents come together in a common nucleus. Fertilization is much slower in mammals than in the sea urchins. The first cell division occurs 12 to 36 hours after sperm binding in mammals, compared with about 90 minutes in sea urchins. Cleavage. When fertilization is completed, a succession of rapid cell divisions ensues. During this period called cleavage, cells undergo the sDNA synthesis and mitosis phases of the cell cycle, but often virtually skip the GA and G2 phases, so little or no protein synthesis occurs. The embryo does not enlarge during this period of development. Cleavage simply partitions the cytoplasm of one large cell, the zygote, into many smaller cells called blastomeres, each with its own nucleus. The first 5 to 7 divisions form a cluster of cells known as a morula, Latin of mulberry, in reference of the lobed surface of the embryo. As at this stage, a fluid-filled cavity called the blastocell begins to form within the morula and is fully formed in the blastula. Blastula, which is a hollow ball of cells. During cleavage, different regions of cytoplasm present in the original and undivided egg cell end up to separate blastomeres. Because the regions may contain different cytoplasmic determinants, in many species this partitioning sets the stage for subsequent developmental events. The eggs and zygotes of sea urchins and other animals, with the possible exception of mammals, have a definite polarity during cleavage. In such organisms, the planes of division follow a specific pattern relative to the poles of the zygote. The polarity is defined by the uneven distribution of substances in the cytoplasm, including specific mRNAs, protein, and yolk stored nutrients. In many frogs and other animals, the distribution of yolk is a key factor influencing the pattern of cleavage. Yolk is most concentrated toward one pole of the egg, called the vegetal pole. The yolk concentration decreases significantly toward the opposite pole, the animal pole. Notice that the animal pole is also the site where the polar bodies of our genesis are budded from the cell. The 3 body axes shown here in this figure, notice that, are established early in development. This process has been well studied, in particular for frog species, where different regions of the egg and early embryo can be distinguished by color and followed easily. The animal and vegetal hemispheres of the zygote, named for their receptive balls, differ in color. The named, notice that, the animal hemisphere is a deep gray because dark-colored melanin granules are embedded in the outer layer cortex of the cell. In this region, the lack of melanin granules in the vegetal hemisphere allows the yellow color of the yolk to be visible. Following fusion of the egg and the sperm rearrangement of the amphibian egg, cytoplasm establishes one of the body axes. The plasma membrane and associated cortex rotate with respect of the inner cytoplasm. The animal pole cortex moves toward the point of sperm entry, and the vegetal hemisphere cortex across from the point of sperm entry moves toward the animal hemisphere. Molecules in the vegetal cortex are now able to interact with inner cytoplasmic molecules in the animal hemisphere, leading to the formation of cytoplasmic determinants that will later initiate development of dorsal structures. Those cortical rotation establishes the dorsal ventral back belly axis of the zygote in some species. This event also exposes a light gray region of cytoplasm, the gray crescent, that is covered by the pigmented animal cortex near the equator of the egg barrier to rotation located on the side opposite sperm entry. The A gray crescent can be used as a marker for the future dorsal side of the embryo. The lighter pigment of the gray crescent can persist through many rounds of cell division. As shown in this figure, notice that. This figure shows the cleavage planes during the initial cell divisions, or in frogs, the first two divisions. These are meridional vertical, resulting in 4 blastomeres of equal size, each extending from the animal pole to the vegetal pole. The third division is equatorial division, horizontal, producing an 8-celled embryo. However, the highly uneven distribution of yolk in the frog zygote displaces the mitotic appearances and eventual cytokinesis toward the animal end of the dividing cells in equatorial divisions. Thus, the 4 blastomeres in the animal hemisphere are smaller than those of the vegetal hemisphere at the 8-cell stage. The displacing effect of the yolk persists in subsequent divisions. Continual divisions produce a morula and then a blastula in frogs. Because of unequal cell division, the blastocell is located in the animal hemisphere. Although the eggs of sea urchins and some other animals have less yolk than frog eggs, they still have an animal vegetal axis, owing to uneven distribution of other substances without the restraints imposed by yolk. However, the blastomeres formed during cleavage are more likely to be of similar size, particularly during the first few divisions. Nonetheless, the general cleavage pattern seen in frogs is seen in sea urchins or other echinoderms in all chordates, and indeed in all animals classified as deuterostomes. In animals whose eggs contain relatively little yolk, to be the blastocell is centrally located. Yolk is most plentiful and has its most pronounced effect on cleavage in the eggs of birds, other reptiles, many fishes, and insects. In birds, for example, the port of the egg we commonly call the yolk is actually the egg cell swollen with yolk nutrients. There is very small disk of actual cytoplasm located at the animal pole. This, as shown in this figure, step number 1, this enormous cell is surrounded by a protein solution. The egg is white, and that will provide additional nutrients for the growing embryo. Cleavage of the fertilized egg is restricted to the small disk of yolk-free cytoplasm and cannot penetrate through the dense yolk. Thus, the yolk remains uncleaved. Step 2. This incomplete division of yolk-rich egg is known as metroblastic cleavage. It contrasts with holoblastic cleavage, the complete division of eggs having little yolk, as in sea urchins, or a moderate amount of yolk, as in frogs. Early cleavage divisions in a bird embryo produce a cap of cells called the plastoderm, which rests on the undivided egg yolk, the blastomeres, and sort into upper and lower layers, the epiblast and hypoblast. Step 3. The cavity between these two layers is the avian version of the blastocell, and the embryonic stage is the avian equivalent of the blastula, although its form is different from the hollow ball of an embryo, an early sea urchin or frog embryo. frog embryo. In insects such as Drosophila, the zygote's nucleus is situated within a mass of yolk. Cleavage begins with the nucleus undergoing mitotic divisions that are not accompanied by cytokinesis. These mitotic divisions produce several handed nuclei, which at first spread through the yolk and later migrate to the outer edge of the embryo. After several more rounds of mitosis, a plasma membrane forms around each nucleus, and the embryo, now the equivalent of blastula, consists of a single layer of about 6,000 cells surrounding a mass of yolk. Gastrulation, the morphogenetic process called gastrulation, is a dramatic rearrangement of the cells of the blastula to form a three-layered embryo, which a primitive gut. Although gastrulation differs in detail from one animal group to another, the process is driven by the same general mechanisms in all species: changes in cell motility, changes in cell shape, and changes in cellular addition to other cells and to molecules to the extracellular matrix. The result of gastrulation is that some of the cells or near the surface of the blastula move to an anterior location, and their cell layers and the three cell layers are established. The three-layer embryo is called the gastrula. The positioning of the cell layers in the gastrula allows cells to interact with each other in new ways. The three layers produced by gastrulation are embryonic tissues, collectively termed the embryonic germ layers. The ectoderm forms the outer layer of the gastrula. The endoderm lines the embryonic digestive tract, and the mesoderm partly fills the space between the ectoderm and the endoderm. Eventually, these three cell layers develop into all tissues and organs of adult animal. Here we examine the events that occur during gastrulation in the sea urchin, frog, and chick. Outlines gastrulation in sea urchin's embryo. The sea urchin pastula consists of a single layer of cells enclosing the centrally located blastocell. Gastrulation begins at the vegetal pole, where individual cells detach from the bastula well and enter the blastocell as migratory cells called mesenchyme cells. The remaining cells near the vegetal pole flatten slightly and form a vegetal plate that blocks inward, a process called invagination. The blocked vegetal